Welcome. We're going to pick up here with the 12.2 podcast as we wrap up Chapter 12. Starting off, I just wanted to make sure, in case we're looking at the microscopes, which we will be coming up, that as you guys kind of look through, you can understand the different phases when you look at them. So interphase will look like it's got a pretty solid nucleus. So if you see it, you'll see the general cell structure, which is oftentimes going to be pretty transparent. And you'll see one darker body inside. That's your nucleus. It's one of the largest, if not the largest, organelles. And then sometimes inside of that, you might see another dot or a couple dots that'll be nucleolus or nucleoli. When you get to prophase, you'll start to be able to see these chromosomes. So you'll start to see where it looks like it's made up more of actual distinct filaments. Those are obviously a lot thicker. It's DNA that's wound up and condensed down quite a bit. But it'll start to look like it's got these pieces. But they'll still be kind of in a ball shape, if you will. Pro-metaphase is almost impossible to distinguish, especially between prophase and metaphase. But if we jump to metaphase, you'll be able to see that they line up at the center. So this is that checkpoint we talked about, while they make sure that all the kinetochores are attached to the centromeres and to the spindle so that they can get pulled apart properly. Anaphase, you can tell pretty easily just because you'll have the chromosomes being pulled apart, and you'll typically see that there's not a whole lot of cytokinesis going on. So the cell looks elongated as those polar fibers that go from one side to the other kind of push, and they start to elongate the cell in general. And then as those spindle fibers and the kinetochores start to retract, literally disassembling generally to tug, and so they'll pull the two chromosomes towards either end, so that way either end can get its own nuclei. And then in telophase, you'll typically see that cytokinesis start kind of dividing this cell up, and you'll, depending on exactly where it's at in telophase, in some cases the nuclei will look pretty much like interphase. In some cases they'll still look a little bit like prophase. It kind of depends on if it's already dismantled the chromosomes, if it's already fully formed the nuclear envelope again. Late telophase will look a lot like a cell that's just about two cells in interphase, but those two cells just might still be slightly connected as they get divided. So that's all that is, just kind of a review anyway. But we do have this whole group of cells that oftentimes get treated like the unwanted stepchild, unfortunately, in biology, and that's the prokaryotes. They're amazing, they can do awesome stuff, but because they tend to be, on an individual level, more simplistic, and because we're not prokaryotic, and because they're so small, because they're all unicellular, and at least in America size matters, we don't like the fact that they're so small. And in some cases they can kill us, and people hold a grudge. So people tend to ignore prokaryotes. But binary fission will be the way they ultimately go through and divide. And because they're simpler, this is actually a much simpler process. They're going to have one circular chromosome, so not too difficult there. They're going to copy that circular chromosome, so now they've got two circular chromosomes. And what this one doesn't show is they typically will anchor this to parts of the cell membrane kind of earlier on in the bacterial life cycle. So as the bacteria grows, it kind of pulls apart those two chromosomes because they're attached to kind of different sides of the cell membrane. So as that cell grows, they get tugged apart until eventually it goes through and it splits, whether it's by dividing if it's one with a cell wall, kind of like the cell plate, or pinching apart if it's one without it. But it's ultimately going to divide in two via cytokinesis, and we will get two new bacterial cells. Because they're simpler, this process is much faster. You can be talking for an entire cell cycle in prokaryotes in ideal conditions. You can be talking on the order of minutes, half an hour or something between divisions if it's ideal conditions. Whereas if you're talking about eukaryotes, you're normally talking about hours or in some cases days. They've got more stuff going on. Imagine trying to move your dorm where you have everything that fits in your car versus trying to move a house that has 8 bedrooms where you're going to have to get and make 2 U-Haul runs to get all the stuff from one place to the other. It's going to take a lot longer if you've got the big house. And so eukaryotes take longer to go through cell division than prokaryotes. This does appear to be a mitotic predecessor, and so what that means is a lot of stuff that's somewhat similar to mitosis happens here, but it's way scaled back because they don't need an elaborate spindle apparatus. They can still use some of those for cell shape to elongate the bacterial cell. They can still use it if they're trying to pinch stuff apart, but they don't need to have all the kinetochores and the spindle fibers because they're not separating a whole bunch of different chromosomes. It's not as elaborate of an operation. And we do have some protists that are kind of intermediate, where they kind of have some of the things that still look more similar to prokaryotic cell division, intermingled with some of the stuff that's more similar to what we think of as the traditional eukaryotic cell division, the traditional mitotic pathway. So we do see some guys that are kind of in between, which helps us connect this idea of binary fission ultimately to mitosis. So there does appear to be this connection, but binary fission, just keep in mind, is way simpler. You're going to copy your DNA, you're going to separate the DNA to either end, and you're going to go through cytokinesis. That's all you have. There's no fancy mitosis steps to deal with. And then we've got the signal checkpoints. We've been talking about signal transduction and cell signaling, and so we're actually going to get back to that here a little bit as we talk about how to control this process. So we've mentioned G1 and how ultimately a cell can go to G0 instead if it says, I'm just not going to do this. I'm not ever going to go through the cell division process. But assuming that you're going to go on, you're going to go through this G1 checkpoint, which will move you on into the S phase. So this is one of the first big checkpoints where you can kind of go either the G0 route or you can go to the S route. You can move on from G1 to this next phase that we would call the synthesis phase, where once again, you're not necessarily going to divide immediately by any means, but you've kind of committed that somewhere in the future you're going to divide, because you go through, you copy your DNA, you start to get prepped. Now this process can still take, which could be a long time for the cell to say it takes hours or days, but ultimately it's still going to happen. You're still committed to the process of cell division. So that's the G1 checkpoint. You then have the G2 checkpoint, that's a major one, and this is where you ultimately commit to specifically divide, not like in the future, like now. That you are going into the M phase, which is going to be followed by cytokinesis that's kind of tucked into the M phase. And so you were going to go through this actual division. So it's kind of like getting pregnant versus saying one day I'd like to have a kid. And this will be controlled by a specific type of cyclin that binds to a guy called CDK, which is cyclin-dependent kinase. So what this means is this kinase is activated by this secondary messenger called cyclin. And so when the cyclin concentrations get high enough, so we can draw little concentration bars, when they get high enough, it'll bind to CDK and it makes a compound called mitosis promoting factor, or MPF. And that ultimately, when there's enough MPF, makes you enter mitosis. So nothing really crazy there. Mitosis promoting factor makes you go into mitosis. And then during the M phase, we brought up that there is a brief checkpoint where you try to make sure all the chromosomes are lined up before you divide them so you don't screw up and end up with a daughter cell that's got extra chromosomes or is missing chromosomes, because that is generally fatal. So we don't want to go through that process if we can avoid it. And you'll see here there are other types of cyclins, so it's not like there's just one. We normally kind of talk about the G2 part here, where we've got this cyclin B and CDK1. I'm not going to make you remember any special type of cyclins. I'm not going to make you remember special types of CDKs, but just know that the whole cell cycle is pretty much regulated by these different cyclin secondary messengers, if you will, where their concentration binds to this CDK, these cyclin-dependent kinases, and activates them, which is what gives the go signal. It says, all right, let's move on. And then regulation here, additionally, we have growth factors and such that will ultimately trigger the production of the cyclin. So you'll have, as a cell gets older and bigger, it might produce some more cyclin, but there's also chemical signals that can trigger the production of cyclin as a means of getting you to go through cell division. So if there's been cellular damage and such, you can just have where cyclin gets released. That then gets you through these checkpoints, which then gets you to go through cell division, which can help patch whatever the issue is. Or if you're trying to grow because you're releasing testosterone or estrogen, like puberty, all those things can lead to cell division by increasing the amount of cyclin as kind of their side effect. So growth factors are one of those things. And then we've got some other things that are kind of odd versions of regulation. So density dependent inhibition is where if cells start to get too packed. So let's say we've got this petri dish that's got a bunch of bacteria or eukaryotic cells, and they're kind of all lined up together. If they start to get too packed, they'll oftentimes, because of a lack of space or resources, they'll dip the amount of cyclin that they have to prevent themselves from going through reproduction. Now, if somebody dies off and they find that they have some space, they'll oftentimes reproduce then to fill in that gap. But so long as they have one row, they're not going to just go in and start packing a second row. They won't do that. And so that's called density-dependent inhibition. So not all cells will follow this, but many cells will use this where they try not to get too populated. You know, it's kind of like when you have rabbits, you might get a lot of rabbits and they take up a lot of space horizontally, but we don't start stacking the rabbits. They don't do that. Now in this case, it's an automatic controlled process. The rabbits, I don't know, they might still try and have babies, but the babies would just die. And then anchorage dependence, this one's where some cells won't reproduce unless they're stuck to something. So a common example of this is you have the urethra, and so this is in men and women. This is ultimately where urine flows out. And there are bacteria, many of them are ones that naturally live without incidence within our digestive tracts. But if they manage to get into the urethra, if they can stick to the side, they'll start to go through this process of cell division. And this is bad for us because when they're stuck to the side there, going through cell division and just trying to live, they're not trying to harm you, but it ends up inflaming the urethra, which is why it hurts to pee and it burns. And it's because you have inflammation in that urethra from these guys binding, because that's their way of kind of making sure that they're safe. They want to bind to something before they start going through the reproductive process. So they try to stick to stuff, and it's just a really bad thing then if it gets into your urethra. You do not want them to get there. And so you'd end up having to get some antibiotic to clear that up. And then the last bit that we have here is if the whole regulation process goes wrong, if the whole cell cycle, the whole cell division, all that just really takes a bad turn. And that's ultimately going to lead to cancer. Now, before I kind of get into cancer, I should say that there's two different types of tumors, two different types of screw-ups. The first one that you'll see is usually called benign. And this is the one if someone says that we found a lump. So if you're looking and they've got, this is breast tissue, so if you've got like this lump, you're first going to cross your fingers and say, I hope it's benign. You know, I hope it's something that ultimately isn't going to be that big of a deal. Because if it's benign, that means that it does tend to have what all tumors do, which is just kind of uncontrolled cell growth. And that just means that it's not listening to our signals. It doesn't necessarily listen if you say, you know, apoptosis. It doesn't necessarily listen if you say, look, we're too packed. You know, density-dependent inhibition, quit. They'll oftentimes just reproduce, and they don't pay attention to the checkpoints. And so this will give you uncontrolled cell growth. But benign is the one that stays put. So these are kind of like the lazy tumors. And so if you end up getting a tumor that's in your colon, that's in your breast tissue, that's in your arm or bone, if it's benign and stays put, it's usually not a big deal. I say that because we can either just remove it, or worst case scenario, which certainly can be bad. Don't tell me I'm advocating this, but you might have to lose like an arm or part of something if the tumor is getting in the way and causing problems. But if it's somewhere that we can remove, you know, if it's ultimately something like in your forearm, you can get by without the forearm. And so it's not normally going to be lethal unless it happens to be somewhere like in the brain where it can put pressure on the brain and it can't be operated on. So occasionally these guys will be problematic. But in general, these will be the good kinds of tumors if you can say there's a good kind of tumor. Now, the definitely bad type of tumor is the one that would be called malignant. And malignant's going to be very similar to just saying cancer. So oftentimes, instead of saying you have cancer, they might say you have a malignant tumor. And the idea with malignant tumors is not just the uncontrolled cell growth, because they have that too, but they also have metastasis. And this basically means that they like to travel. And so they like to go elsewhere in the body. So breast cancer will never, that I'm aware of, possibly kill you by just being in the breast tissue. Just like you'll see that a lot of other cancers won't necessarily, like skin cancer, melanoma, one of the deadliest cancers, won't kill you by making your mole turn funky. You know, that's horrible, your mole's funky, but that's not the danger. The problem is that a lot of these ones like to go to your lungs, they like to go to your brain, they like to go to your pancreas. And if they go somewhere that we can't operate on, that we can't easily remove it, that you don't have extras of, like a kidney, or you could perhaps give up part of your liver and it regenerate. So if it's somewhere that we can't mess with, that's what can ultimately cause you to die. Because these cancer cells will kind of crowd out the healthy cells. That's their issue. And if they crowd out the healthy cells, they make it where the actual kidney or lungs can't do their job. And if your lungs, your kidneys can't do your job, you end up with organ failure, and that can lead to your death. So it's not like the cancer cells directly go and start killing your cells. It's more just they get in the way of all your cells because all they're doing is eating and growing, and so they keep taking up more and more space and getting in the way of all the vital functions that you need to get done. And this is going to happen because there's some type of transformation, oftentimes a mutation in your genes that breaks all these normal signals. And so it's going to break things like we talked about, like the cell signals. It breaks essentially these checkpoints. And so without our ability to control, to have oversight over cell growth, you can rapidly get where things get out of hand and they start to become essentially selfish cells. They're not just being normal somatic body cells that do their job. They're all about just trying to get resources and divide. And in the process, all of our cells in a multicellular organism, or at least most of them, need to do their job for us to live, and they prevent that from happening. So that's why when you go and you have like a biopsy because they fear that you might have something that's a tumor, that's why you see them talk about and kind of sitting there hoping that it's benign instead of malignant.